Huh? Okay.
This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
It's Paul Verschure with the Convergent Science Network podcast, together with my colleague Tony Prescott, and with Murray Shanahan, who was speaking this morning in our summer school here in Barcelona. And Murray, you presented, let's say, a bit more an abstract view on cortical dynamics and how we could think about cortical dynamics. So what are the key features of this model of cortex you're presenting, and what are you trying to explain?
So I think the key feature of the model, which reflects my current interests really, is the richness of the dynamics that it produces. So metastability was one of the main themes that I talked about this morning. So that's one important feature, which is related to the size of the repertoire of different states that the system can produce as well. So metastability, just to spell that out. So a system is metastable. Basically, if it instead of falling into a stable attractor, it sort of lingers in the vicinity of an attractor-like state, without stabilizing and falling straight right into that attractor, and then maybe moves on to a different attractor. Mm. So for listeners for whom that isn't very familiar terminology, so the idea would be, or one kind of attractor state would be where all the parts of the brain were all oscillating in synchrony all at the same time. And of course, this never happens, and it would be very, very bad if it did. But that's one kind of attractor state that you can get in dynamical systems, is where you've got a whole load of oscillators that are all completely synchronized with each other. Now, in the brain, I think the kind of dynamics we see is where there's lots of oscillations going on. There's lots of patterns of synchrony. But what we're really interested in is states of partial synchrony, where some parts of the brain are synchronized with other parts of the brain. That often represents that they're talking to each other. And we're also interested in metastable states of synchronization, so where it doesn't stay in that exact combination of synchronized parts, but that sort of coalition of synchronized parts is like that for a while, and then the coalition breaks apart and it finds another state.
But now you could argue that this metastable state, which is defined in a state space, is in some sense arbitrary, because as observer, I can decide at what level I define my state space, and what's metastable at one level of description can just be a fixed point attractor at another level of description. So do you see that as a problem, this arbitrariness of how we define such a state space?
It certainly doesn't have to be so arbitrary, and there are various ways that you can try to quantify it precisely. So one way, for example, is to look at, in the case of these coupled oscillators, if you have a whole collection of oscillators that are decoupled, then there's no connection between them at all, really. And in that case, that's the sort of the trivial case where there's not going to be any synchronization except coincidental synchronization. So that's the statistical baseline. So if you're looking at your system and things fall into synchrony, a lot more than you would expect them to, in the case of that kind of baseline statistics, then you know that's serious synchronization, serious sort of
but
metastability.
Are you saying with this metastability concept that, let's say, between stability and complete chaos, it's in this intermediate zone that interesting things are happening?
Yes, that's certainly very
like
much
an edge of chaos kind of.
It's very much related to that. Yeah, very much related to that. So there is a kind of criticality phenomenon there, where the sort of systems that you're interested in, or the dynamics that you're interested in, is poised between order and disorder. So a highly ordered system will be one that was just, say, completely synchronized, and a highly disordered system will be one like the one I just described, completely decoupled. There's no relationship between the different oscillators. What you're interested in is this state which is between the two, where there's a certain amount of order and a certain amount of disorder. So the parts are sort of interacting with each other, but they're not totally, there isn't one dominant state that persists.
So now you were defining metastability also in relation to the physiology of people like Pascal Fries, who comes from the school of Wolf Singer, who sort of ascribe a lot of importance to, let's say, synchronization phenomena in the brain.
Yeah.
Right. So how are these two things now related?
Yeah. So that's very much the backdrop for the discussion we've just had. Really, is so Pascal, according to Pascal Fries's communication through coherence hypothesis, which makes a lot of sense to me. You can think of two populations of neurons that are oscillating in synchrony as basically in a position to communicate with each other and cooperate with each other, to influence each other and exchange information and so on. Because when the troughs and peaks of their activity coincide, then they're in a good position to exchange spikes, so long as all the delays work out and so on. That's the reason why synchronization is of one. It's a hypothesis, but it's one reason why synchronized collections of neurons might be of interest if you're looking at populations on different parts of the brain.
Counterintuitive, because in some sense, if I now would look at these two neurons and I would plot their activity over time, but against each other, they might actually be in some sort of fixed point. They might be in a very limit cycle, but it would not look like a very variable metastable state. So how is then the coherence in gamma reflecting this metastability or this criticality phenomena?
Yeah. Well, first of all, you've got to look at different time scales. And you're also looking at whole populations of neurons. So you're certainly not looking at just single neurons, but you're looking at populations of neurons. So you might look at one population of neurons. You might see that the overall firing rate, the mean firing rate, is oscillating for a while, is oscillating in a regular kind of way at say a gamma type frequency, 40 hertz or something, and then you've got another population that maybe is also oscillating in the same kind of way, and the oscillations are in synchrony, and then those two populations are in a position to exchange information, and so that's the kind of situation that you're interested in. Now, the information is going to be exchanged during those little peaks of activation. And during a peak of activation, then that whole population is going to be most receptive to incoming spikes, and is going to be, the individual neurons are going to be more likely, statistically, more likely to fire than in a trough of activation. And so that's why the communication can take place, in that time.
But surely if you're thinking about maximizing the opportunity to fire the postsynaptic neuron, you don't want to come when it's firing. You want to come just a bit before that.
Yeah, okay, that's absolutely true. But you've within 40 hertz, you've got quite a few milliseconds. It's a little window of opportunity. Right. So you want, the timing's got to work out. That's for sure. You've got a window of opportunity. So quite a lot can happen in that little window. And also you've also got to worry about the delays. So there are going to be delays between depending on the length of the connection and the type of connection and so on. There are going to be delays as well. So all of that's got to work out. But the point is that it's getting the right relationship in the phase to work out is going to maximize the opportunity for information to be exchanged, and also allows for competition. So you might have three populations. One population is two populations are trying to influence a third, and one is going to win out by entraining the downstream population. Right. And once it's become entrained, then it can shut out the other guy who will end up in the opposite phase relationship, and that's when the information sort of.
But now the idea about gamma, the dominant idea about gamma, is that it's very much a locally generated response, right? So it's found, let's say, cortical circuits or in the hippocampus. It's actually the tight coupling of excitatory neurons with local, fast GABA-A mediated inhibitory neurons.
Yeah.
So as soon as you start to drive the excitatory cells, they will drive the local inhibition that will shut down the whole population. So now you get a rhythmicity in the gamma range, right? So you could then first argue, okay, if I have a gamma in two areas, it means, okay, there's a drive onto these cells. This is the first thing, right? And then you can say, well, now if these two areas talk to each other, okay, by necessity, it will happen within this gamma rhythm because they cannot fire at any other rhythm, but they become driven. Okay?
Yeah.
So then how meaningful is it really to talk about, let's say, the enhanced communication between areas when gamma becomes more or less coherent?
Yeah. Well, I think it's particularly meaningful in the context of this kind of competition. So where indeed you may have several neuronal populations are all, as it were, relevant to the ongoing situation. So if you've got an example, a potential example is binocular rivalry. Maybe you know about binocular rivalry. So where you've got two gratings, one horizontal, one vertical, presented to each eye. And so the right eye can see the horizontal grating, and the left eye the vertical grating. And then there's this binocular rivalry phenomenon whereby you don't see the two gratings overlaid on each other. You don't see a crisscross pattern, but rather you tend to see consciously you'll become aware of either one or the other. And the one will fade in and out while the other one fades in and out. So that's known as binocular rivalry. And so that's an example of where there's clearly some competition going on between rival neuronal populations. And so one way possibly to account for that is in terms of this communication through coherence idea, where one population temporarily entrains another population further downstream, and that dynamics goes on for a little while, and then it can sort of burn itself out for statistical reasons, basically, and then because they're equally salient stimuli, and then the other population becomes entrained instead, and so you have this flickering in and out.
Right.
And we had a paper in Frontiers in Computational Neuroscience paper. I had a paper with one of my PhD students, Mark Wildey, where we showed that kind of phenomenon in the context of Pascal Friese's ideas.
Yeah, so now we know a little bit, let's say, the physiological phenomena we would like to understand and explain, right? And what you told us is that actually you start to model these large-scale network phenomena with fairly abstract oscillator models.
Yeah.
So why did you make that step?
Yes, I think I made that step just because I just fell into it. It wasn't really that I took a very careful decision about what I should look into next. But I mean, my background is in computer modeling, so my natural tendency is to tinker with my computer models. So I'm sitting there with MATLAB, fiddling around and playing with my computer models, and that's what I do.
I love your job.
Yeah, but I'm always vulnerable to there's so a lot of people are interested in that kind of thing in its own right. Physicists in particular, kind of quite interested in that sort of thing in its own right. But then if you're trying to make a claim about its relevance to brain dynamics or cognition or anything like that, then of course you're instantly vulnerable to the criticism that, well, where's the data? How does it relate to data? And so of course it's very important to try and relate it to real data. But in my case, it's been a bit back to front. So I was tinkering around with the models for a lot, and only a little bit later did the opportunity to make it match up with real data come in.
So what are the key parameters that you then fiddle with when you're sitting behind MATLAB on your desktop computer in your office?
Oh, well, so in coming up with that particular model with the metastable chimera states, I tinkered with so many things, but particularly with the network construction. The sparsity of the network, lots of parameters to do with the network construction. The parameters of the oscillator models, the coupling. I mean, there are hundreds of them. And of course, if I was a good mathematician, I'm handicapped by being a mediocre mathematician, by not dealing with real data, not being a proper scientist. The only thing I'm any good at is programming. So you end up tackling it is like Dorothy just now was talking about. If you're capuchin and you see you've got a hammerstone, then you see everything as a nut. Well, it's a little bit like that with programming. I see everything as a programming problem.
Right.
So I tend to.
But then for the model to close that, I think the key parameters you're controlling is, let's say, it's the phase of your oscillators. I guess the transduction delay in their interactions, the strength of interactions.
Yeah.
These would be roughly the key parameters that would be dominating the properties of your network.
Yeah, so the coupling, so the two main parameters that we end up with fiddling around with are the coupling and the delays.
Mm-hmm.
Yeah.
Okay.
Yeah.
I mean, I'd just like to go into those models a bit more, because one of the things that surprised me in your talk was, I mean, you described some of the history of the study of oscillators, which has been going on for hundreds of years. But then there's some fairly fundamental discoveries that you have made, and I think you mentioned a 2002 paper about properties of oscillators that we didn't know about. And can you say a bit more about those discoveries? Why didn't we know those things before? And is there much more to know about these simple oscillator systems? I know there are large numbers of oscillators, but there seems to be potentially for areas like brain science, lots of scope for more discoveries in this area.
Yeah, I think it's because it's physicists do all the serious legwork with all the heavy lifting with all of these oscillator models, and they have a particular mindset which tends to focus in on things like, let's look at the limiting case where there's an infinite number of oscillators, for example, or let's look at the total, where everything is completely connected, and also let's look at stability conditions. Let's look at all these bifurcation diagrams and the stability conditions. And in fact, there's kinds of dynamics which I'm interested in. I don't think they're actually very surprised that they exist. I just don't think they particularly thought that they were interesting. Is what I suspect, and it's only. But I mean, they seem to be getting interested, because that particular paper of mine has been quite well cited by physicists in looking at it. I mean, on a relatively speaking. Mm-hmm. But I think it's partly because they probably knew that these sorts of things went on, but they weren't interested in irregular networks that are not homogeneous and.
But could you then elaborate a bit what you discovered with this model, the so-called chimera states, and why they are interesting?
And perhaps also how they relate to different network configurations, because that's obviously a key parameter.
Yeah, sure. So why they're interesting is,
maybe first define what they are.
It. So these chimera states is where you have a set of oscillators that basically partitions into two or more subsets, where two subsets really, where one of them is synchronized and the other is desynchronized. So that's a chimera state. And when these were first discovered by the physicists, I think they found it quite surprising that these things could exist, especially because they had very homogeneous connectivity in those ones that they were looking at. So they were a bit surprised by that. This is certainly of interest from the perspective of brain dynamics, because you're not really interested in states either where there's no synchrony, because nothing's happening of interest at all there in the brain, nor are you interested in the case where everything is synchronized, because that's basically seizure, and nothing interesting is happening either. You're interested in the case where some populations are synchronized and some are not, and the ones that are synchronized are the ones that are governing behavior. So, we should try and relate this maybe a little bit to the behavior of animals, right? Because we've been talking very abstract terms.
No, but I think the key thing about your chimera states was also, you see it as a signature of, let's say, a network property, right? Where you would say, well, now I start to get minorities, if you want, in the whole population of oscillators that start to, let's say, be out of phase with the majority.
Yeah, that's right.
This.
Well, actually, I think it's the minority that are going to be synchronized, and the majority are going to be because.
Oh, your data looks a bit different, I must say.
In the data, it's in that particular model, it's usually about half and half. Actually, okay. So about half in that particular model, about half the oscillators end up synchronized with each other, and about half end up desynchronized. But I think in real data, you probably expect a smaller set to be synchronized and to be governing the behavior of the animal at that time.
That's also the link to your metastability, right? Where you say, look, we want to be between order and chaos. which would roughly be expressed in such a network as these chimera states. So you say, look, now I am at this critical point where interesting stuff can happen.
Yeah,
right?
But it's not just the chimera state; it's the metastability, because you don't want to be stuck in one chimera state either.
Sure, absolutely.
You want that particular coalition of synchronized oscillators. You don't want that to be the same one forever. Mm-hmm. It's doing its thing for a while, and then you expect it to break apart, and then another coalition of oscillators, so a different chimera state to arise.
Exactly.
So, but you've got this minority group of coupled oscillators, and then you say these ones
synchronized
which are synchronized. These ones that are desynchronized, what do you think they're doing? Are they just playing along in the background but not having any influence?
I think that's exactly what they're doing. So,
any evidence for that?
Things that are not synchronized or not even exhibiting regular oscillatory behavior. I mean, that's the mosely what's going on in the brain, right?
But I mean, I wouldn't want to go as far and say that things that aren't oscillatory aren't of importance, and most of the sensory events aren't oscillatory.
Sure, that's true. So I think, I see it as, but I mean, this is a hypothesis that demands empirical validation, but I see it as a mechanism for communication and cooperation among populations that are anatomically distributed around the brain.
Right.
And so I think we should talk a bit about behavior, right? Because all of this has been very abstract.
Behavior, when you're saying, okay, the interesting stuff happens in the synchronized oscillators.
Yeah.
Is that because you have some fundamental theory about brain hardware that, perhaps goes beyond this information transmission thing? I mean, so what's happening in the oscillation is information is being passed across the network, but these other decoupled ones are still processing information perhaps and passing it around. And I'm just not clear why that wouldn't be important
I'm not saying it's not important. Okay. I mean, I'm very much, against the idea of saying there's one mechanism to rule them all in the brain, because everything, I come to the brain as an engineer, and I'm constantly trying to do that. And it's because that's my background and training. You want some kind of set of engineering principles that you want to squeeze the brain into. And I'm constantly being brought up short and made to realize that no, in fact, what you thought was a principle is only a statistical tendency. And in fact, there's much more of a mess there. So I think this is one mechanism that may be quite important, in the brain, but I wouldn't want to say it's the only one by any means.
Right. I was just wondering if there's some kind of thing about clock speed or whatever going on in the brain here that you think might be interesting.
Well, I think there is actually, as Paul was saying earlier on, there is actually an inherent tendency for these systems to enter oscillatory regimes when they're driven. And when you build models, you see this pretty quickly. You build a model, let's say, with spiking neurons, and the excitatory connections there's a lot of excitatory connections there, and you drive it, it's going to naturally start to oscillate. And if you've got broadly biologically accurate parameters for your spiking neurons, it does tend to oscillate in around about the gamma frequency. It just does that. So that probably falls a little bit out of the neurobiology. Out of the biology. And then I suspect that the brain is going to find, I think whatever rich possibilities for dynamics you've got in the brain and evolution are going to find some way of exploiting them. Mm-hmm. So you've got these oscillating things going on, and the brain is going to find a way of exploiting the potential of this. And I think one way is that it facilitates competition and cooperation among anatomically distributed populations.
But that raises on the next question, right? Whether these chimera states would also just drop out of the kind of local circuitry of cortical networks in the same way gamma, you get it for free, or whether something else is required for that.
Right? That's a very good question. I suspect that they probably do drop out. But that's a very good question. Mm-hmm. It would be good to construct models that prove that. I mean, one reason why they might drop out is because another thing I've noticed is that in competitive mechanisms, you try and build some kind of competitive winner takes all mechanism. I mean, Tony might know about this as well, but you've got to build it in a very particular kind of way to make the winner persist, really persist indefinitely. And it doesn't take much, especially if you've got small populations of neurons, for a bit of statistical variation to flip the winner to the other one. If you've got two, you've got two rivals. And so that's, again, I think is a natural property. So that does lead to pretty naturally to these kind of chimera states and flipping between them.
But now the next thing you did is essentially you took like the state of the art, if you want, about on human brain connectivity, like the human connectome and its different statistical properties. And we had an interesting discussion about how to interpret that this morning, but we don't have to get into that. But so if you now combine these dynamics and the notions of criticality with our understanding of the human brain, right? So you built these kinds of models. What happened?
Well, so the credit really has to go to the people from here in Barcelona who did this first of all, Gustavo Deco's group and Joana Cabral, who built a model based on the Hagman dataset, where they used the Hagman connectivity dataset and basically put one of these oscillators, Kuramoto oscillators of the type that I was playing with, one on each of the nodes in Hagman's network. So it's very, very similar to the thing that I built, but the thing that I built used a completely synthetic network. It was a synthetic network that had some brain-like features, such as modularity, but it was just constructed by an algorithm. Now they did it with a real DTI-based data set, and then the interesting thing was that they showed that they were able to produce a strong correlation with resting states fMRI data. And that, I have to say that it did rather surprise me, and it pleased me enormously that it only happens when it's in this metastable chimera state, exhibiting this metastable chimera state dynamics. So the kind of dynamics that I had found in my model, and I nobody really had published anything showing that Kuramoto models could produce this. Although, as I say, I think the physicists probably knew it; they just didn't think it was interesting enough. Right. But the amazing thing was to find that somebody could show that it was could be used to model real data, and only if it was in that regime.
But I couldn't. I would like to push you on that a little bit more, because you could argue, look, to get to something like the dynamics of a resting state network, the key thing is that you get your connectivity defined. So then, in this case, you take this notion of functional connectivity, which is a statistical structure of interactions. You exploit that to seed the topology of your network. And
but in this case, it's structural connectivity. So they were using a structural connectivity.
So you have your structural connectivity, and you start to drive that now. Couldn't you argue that any kind of activity model, even a rate-based model, a linear threshold unit, would be able to give you a resting state kind of pattern, because the secret sits in the connectivity and not in the dynamics of the nodes?
Yeah. That may well be the case, and indeed, other people, including again Gustavo Deco's group, have used different kinds of dynamical models. But few of them are simpler than this Kuramoto model. I mean, it's about as simple as you can get and produce, interesting kinds of results. I think so. That's what strikes me.
So the data set that you're looking at there is fMRI, which of course doesn't really have the temporal resolution to look at things like oscillations. So, I mean, can you unpack for us a little bit how that data really validates the model? Because obviously you're looking indirectly for evidence for the kind of states that your model is creating.
Yeah. So we're looking at so in this kind of work, you're looking at how the model over quite a long run. So the model itself is oscillating at a gamma frequency, but it's producing phenomena at a much slower. It's producing dynamical phenomena at a much slower rate as well, where large groups will synchronize for a while, and then that synchrony will then go away. So that is happening at a much slower rate.
So there's a slower dynamics which you can then look for.
So there's slower dynamics, yeah. Right. So right. The actual model is constructed at the level of this faster dynamics, but the results that you produce are at the level of the slower dynamics. So that's where you're making the matches.
And what are the phenomena at this slower rate that you're matching on?
So, you'd have to ask my co-authors exactly what's going on.
No, but there's an issue here, right? Because as you know, there's quite a discussion again now about what are we really measuring with fMRI.
Yeah,
right?
sure.
And there's some correlation with blood flow, but actually the link to neural activity is still being debated, right? So now, at best, with your oscillator model, you capture, let's say, the hemodynamics that is sort of indirectly reflecting something neural that we don't really understand yet. On the other hand, the origin of your model was neurophysiology, a very different level of description, both spatially and temporally, because now we're talking about single cells doing things, right?
Yeah.
So isn't it quite a stretch to actually first say, and I capture Pascal-Fries's synchronization, gamma range communication data and explain it.
Yeah.
And on top of that, I also give you now here the whole human fMRI.
Yeah. Well, actually, the origin of the model is a little bit higher level than that. So you're really talking about populations of neurons. So what you might maintain that one oscillator represents would be the activity of quite a large population of neurons exhibiting a gamma type dynamics.
Right.
But I have to say, I completely agree with you. It came as a total surprise to me that you could use this kind of model in this sort of way, and that it would actually match the data.
But your closest link with the data is more at this fMRI level than at the neurophysiological level, where we look at direct cell.
Well, that's the
electroactivity.
Well, indeed,
right?
but that's only because those are the only experiments that people have where they've tried to make the match, as far as I'm aware. So,
okay,
I'd be very interested indeed to do it at a lower level.
Right. But now the other thing is that if you go for the whole, let's say, populations, where we talk about, let's say, cubic millimeters of cell volume that we're measuring with fMRI. So if we describe that with an oscillator model, then what we're capturing is the activity of millions of neurons of very heterogeneous types. And that actually also the dynamics of these neurons might be much more heterogeneous than your model captures. Like if you look at gamma and say, okay, gamma is locally driven, right? You might actually have all sorts of subpopulations of cells and all sorts of complex dynamical relations, metastable or not, that you're completely blind to.
Yeah, absolutely.
Right? So how are we going to cross that bridge now?
Well, I think as I said quite early in my talk today, that I did actually start off with spiking neuron models and showed how these spiking neuron models produced a kind of dynamics that I was interested in, or complex dynamics that I was interested in. And people were saying to me, "Oh, why do you need all the complexity of these spiking neurons? Why not do it with a simpler model?" So I thought, okay, I'll give that a go. And so I went up a layer of abstraction and moved to these oscillator models, and kind of got drawn into that because it's a whole world in itself.
Right.
But I do very much think it's a good idea to go back down again and to build larger scale spiking models than the ones I was doing before. And I have a PhD student, David Baumick, who's done a lot of work, and there's a PLOS paper last year that describes exactly that. So you've got a much richer variety of phenomena, in fact, with lots of different frequencies interrelating in different ways.
Right, exactly.
Is there some possibility that you might see metastability at different spatial scales in the brain?
Oh, absolutely. In fact, I would be astonished if you didn't.
Right. So here we are. You seem to have a model that's actually pretty powerful in capturing data and describing and interpreting some of this data on the brain. And now you're also applying that to sort of pretty extreme states in which we can push your brain, like under psilocybin.
Psilocybin, yeah.
Sorry. So, so how has the model helped you to understand the dynamics that you would induce with magic mushrooms?
Yeah. So there's. Can I give a little bit of background there? So one of my colleagues at Imperial College, Robin Carhart Harris, has done some pioneering work with psilocybin, which is the active ingredient of magic mushrooms, where subjects are administered with a psilocybin preparation, and then you do functional MRI on the subjects, and you can interpret the results. In fact, you can interpret the results as an increase in metastability, and which in turn you can interpret in terms of a larger variety of states being produced, quite so flipping between a larger variety of states. So you can kind of explain some of these results, some of the dynamics that you see under psilocybin as moving to a more extreme version of the metastable regime that was identified in this model. And Robin Carhart Harris is quite amenable to this kind of interpretation, and we had a joint paper in Frontiers describing that kind of interpretation. And but I think I think a lot of, and so we're trying to apply the model to that as well. But I think that it's very much work in progress, and we have to. In fact, I think the application of this whole methodology is very much a work in progress, and we have to. Refine it a good deal before it becomes really accepted.
Okay.
You said before you wanted to bring this back to behavior, and one of the ways that I expect you will do that is through the work that you've done on cognitive architecture, which is thinking much more about function of brain circuits, particularly cortical circuits. So can you say a bit more about, first of all, the ideas of cognitive architecture that you're interested in and how that might mesh with this work on oscillators?
Yeah. Well, I one of the really fundamental problems that I've been interested in for a while is how it is that a novel coalition of processes can sort of coalesce out of nowhere to deal with a situation that's never been encountered before, and it's clearly. I think that's clearly at the root of human level cognition. Well, actually, maybe even some animals, some non-human animals can do this as well. So I see the rich dynamics that I've been talking about here as a way of maybe addressing that kind of problem. And it also relates to the kind of cognitive architecture that I've been interested in, where so I've been interested in global workspace architectures. You can relate global workspace architectures to a certain kind of connectivity in the brain, where you have a connective core of hub regions, which perhaps can facilitate coalition formation, in particular the formation of novel coalitions to deal with a new kind of situation. So it's this formation of novel coalitions which I think is quite difficult to account for, and that's the kind of thing that ultimately I'd really like to be able to model.
So the connective core that you're talking about there is the bit that's actually active when we're not doing anything, which as external we seem to be thinking rather than attending.
Yeah,
and is that right? And so what kinds of activities that humans do you think we're going to understand better through this?
Well, it's probably the kinds of activities where you're confronted with a novel situation, and you have to pause for a second and sit back and think and stare at it, and then suddenly, aha! You've a solution has come from nowhere, and so I think that probably is engaging a sort of mode of a dynamical mode that enables processes to talk to each other and form a novel coalition that otherwise they wouldn't have been able to do. And I suspect that they can do this via this sort of these centralized regions in the human brain.
But then we still don't have behavior. So
we have a kind of unobservable behavior. So you've got reflection.
Well, that's the point we want to get to. What's the overt behavior we're going to then explain with that, right?
Yeah. Well, I mean, I say I was caricaturing a little bit by saying you sit back and you say aha. But I think you've probably seen this famous video of Betty the Crow bending the hook for the first time to retrieve the bucket. And of course, you can ask questions about that experiment because it's hard to reproduce that kind of experiment. But if we take that bit of behavior at face value, I think something interesting is going on there. Or when children solve the same problem, because you can reproduce that they'll play around, and maybe there's maybe you'll see this physically, but maybe you won't. But there'll be a little moment when their brain is when maybe they actually pause, but their brain is going to go into a mode where a new combination of processes can come together and then it's going to lock onto that. Somehow the brain, as it were, knows that that's the right solution. That's the sort of aha moment. Right. It locks onto that possibility, and of course, it's going to do it straight away. And that phenomenon in the brain, I think, is key, absolutely key to understanding human level cognition. I think it's as important as insight. It's as important as language, I think, and if not more important than language. And that's what I'd really like to be able to understand and capture in a model.
Okay. So then, so Murray, being an engineer and trying to understand the brain using these kinds of models and actually covering quite a range of phenomena, what would be Murray's law that we have to adhere to study the brain?
I don't think I'm entitled to coin
you are
a law.
Now. You're now.
I think it would be play. One word law. Play. Play with your models and make your models play.
Okay.
I think playfulness in as a scientist and engineer, and playfulness in the models. They're, in a sense, they're the same thing, right? We're being creative, and the root of creativity and the creative things we want to create is playfulness.
Now, the other thing is that Tony and I are trying to control the future. So that's why we have Tony check the predictions people make. And since he can just take a train from Sheffield to London so we can save money that way, the question is 4 years from now, Tony's going to come visit your lab there at Imperial. And he's going to say, look, 4 years ago in this podcast interview, you made this prediction, to them checking whether it was validated. What's the one prediction, concrete prediction you could make today?
I think it's that I'll have no more funding to address this issue than I do now.
Good.
Well, we hope that isn't true.
All right, Murray Shanahan, thank you very much for this conversation.
I was queued up for that question, but hearing you answer the previous...
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.